all fine. And then you would just come back to the folder and select the next presentation. This one here, which is Google Slides, this is the only one that's a bit confusing. So when you,
okay.
Yeah, when you click this, it will open up the browser window. And then
I go to
it's online as well. So you might have to wait for these things. It might take some time to load.
and then
And then you
slideshow.
hit slideshow.
Yeah.
What I will also do is I will leave. I already have it here open. So if you remember, you don't actually have to double click on that. You could just come down here to the Chrome browser.
It's already
It'll be
there.
there, and then you can start it.
No problem.
I think that's
Pretty much it.
that's it. Yeah.
Thank you.
If there are a lot of questions for one person, and they run out of time for questions, then just say, "Oh, you can catch them over lunchtime to ask more questions."
Yeah, great. Just to keep the schedule going as
Yeah,
well.
it's important because with lunch it took quite a long time for everyone to get their food, so we don't want to make lunch too short.
Yes.
It'll be difficult.
Also for today, I told the caterers to separate the lunch in 2 places, so we have shorter queue.
Oh, okay, it's a great idea. All right, I'll get the first talk up. So that we're ready. I think maybe I already had it. We can start this one.
And the session that I'm hosting is the first session that I'm listening at this entire event, because I didn't have a chance to listen to anything. And I that's so sad, because I really wanted to get to know a bit more what everyone is
They're all
doing.
recorded.
Yes, so definitely in spare time.
Yeah.
I have on the 29th, I have a 17-hour-long bus drive to my next field work. So that will be the perfect time.
You sorted.
Yes.
Yeah.
I will have no
We'll
other.
check it's recording. Yeah, it is recording. So we're all good.
Everything okay with the audio?
Yeah, I connected on my phone. I could hear us talking. So yeah, so it's fine.
Let's see if everything's fine downstairs, and we're good to go in about 10 minutes. Do you have time for coffee?
The previous session.
Svim oko. Czy budeš li to ili?
Tu sam. Odakle ovaj lik?
Ovaj.
UK. Dobro.
Nešto, UK Institute for Biology.
Ne, zato što ih ima iz svih delova sveta.
Imaju, da.
I mnogo.
Ne razumije sam.
Dobro, ne znam ko da razumije. U svakom slučaju, kad ih vidim dole s onim na nosu i šta ne sve.
A vi su
I što smrdeš u se Ali nije prvi put. Stvarno nije prvi put. To što su dolazili iz cele Evrope, da, stranci kad dolaze, oni iz Downstrada. Dolazili su neki, ne znam, digitalni nomadi, putnici od svake sorte i kad dođu, ja sam se širao kad sam vidio par likova da nose belu kapu, belu majicu i dole papuče, jadranke i šorce.
Ćemo upravo konferenciji izuvali, isključavati.
Pa da. Bila je jedna konferencija kad su izašli, kad su spavali tu po travi.
Da, i sedeli su i uče.
Jeste li bilo malo, bilo malo ne cela sala da je otišao tamo da sedi, ja sam popisao zbog trave, sve su se izgazali. A mi to debelo plaćamo, da je izgleda ovako kao stavljeno. Ali dobro, ba stvarno su čudilo.
Nemo, već su oni samo pomišljeni, ali...
Uopšte nemam problem s tim kod ne smrde. Baš ja zbolim sa tim, znači ovo se pomerim ne mogu da... Najosnovna je stvar da se pokuš prema što lično posao. Da je to tumač.
Za njih, ne znamo, neće da plate, oni se znaju da treba da plaćaju, sve ove ručice, to što imaju. I onda im mole im da plataju, on mora da ih uči. Onako po imenci da ih uriš, da im priđeš da im kažeš nisi platio. Pričaju, trenutno da što preste keš, mora u dinarima, ovo, ne, oni dođu i do you have a Revolut? Da te aprovaram. I ja pupek. Kako vam pupek, ženo, što su ti rekli 1000 puta u kešu. A jedna je rekla, kaže, a mi trebamo da platimo. Jer ova sim kao refundiran, kao ne znam, neki djel ili nešto, ovaj Engles će da to odredi koliko se refundira. Jer on je neki glavni za sve. I onda opet ti mora Da platiš da bi ti refundirao.
Bukvalno se plaća kotizacija za velike konferencije i odbor.
Da. A ne, ali Cure tamo.
Znaš šta, predstavljaju kao da su mnogo, ne znam, kulturni i ovakvi, onakvi, a suštinski je potpuno obrnuto, imaju tu dečiju kulturu, dečiju tradiciju. Meni bi zaista sramotno bilo da se tako pojavim negde. A najče mi je bilo prošle godine kad su dolazili Kinezi. Ja sam se...
Prošle nedelje što
su bili. I, pa da, to što kažeš, bili su više navrata. Prošle godine što su ih iskrcali 50 njih izbjegalo autobusa, ili su pravac gore otišli u Pip. Pola od njih, a pola su se, ne znam, zgadne, pisali. Našli ja gore kad su otišli, sad mi sedimo na sastanku i Kineščići ispred mene i čačkaju nos. Potriguju. I onda sam posle kopirao, to jest pitao sam zapravo, pa su mi objasnili da je to oko njih problem.
Da, pa ja isto. Katastrofa su se.
Na sve sam gledao da potrigujući, to nije jedan bio, nego jedan više puta i ostalo. I onda mi je bilo zašto neko?
Prošle nedelje su bili katastrofa, jer cijela sauna pod zapuštila se. Jednostavno. Ne znam, što možda se dišaju, pa što smo bili prošle. Kako majica saradnju i ona žena pitala ima, kaže, 30 učesnika, zato ima po 8 do 10 minuta, jer oni imaju govor na kineskom, govor na srpskom, pa prikazuju neki dan, od prebili pesmu i igre, nešto. Zamajavaju se. Mene kaže, imamo kreću u 10:30 ujutru sve. Znači, da treba da već bude sve spremno da krenu. Da li one mogu u 9 ujutru da dođu da probaju svi? I mi kažemo, ali fizički nemate dovoljno vremena da probate, jer to treba se postaviti, to nije samo. I ono kaže, ok, i uzme piše direktor u mail.
Dobro. I šta je rekao direktor?
Direktor rekao, pitajte kolegima svi ovo ne. Ali svakako ono što ti to ne. To liči da ti pišeš direktor u mail, a rekla smo ti da, mislim, to nije ti, ja ne znam da je bilo ponekno, što nije fizički moguće, ne možda ti probaju 5 čoveka i da ostavimo kao šefa. Ali su mnogo, i ne znam nešto zastave došla, pa su, Đole je morao da ne vede bili da kače zastave, da ona je igrao kod kamere u konferenciji.
Dobro,
tu, znači, ne kačuje one zastave, pa ona sutra donela druge zastave, kao ipak skinite ove da zakačemo i zakačemo. Onda ona kaže, nije u ravni, treba da bude u ravni, zgodnje stave, a nisu iste veličine zastave. Onda, ako staviš u ravni, onda jedna izgleda malo manje. Pa nije na sredini, da nas vise, da je sviđaju se, mora da nisu da kamera da bude jedna.
Kako ih je zakačio?
Sa onim šenom. Tako da, imali smo baš muku da ih sve sposobno da 5 puta smo sili da ih kače zastave.
Tamo ima 3 metra tu.
A što stvarno ne mete, vi ne držimo i kačemo. Ali 5, znači 5 puta kad idemo i ono kampiri, a ovo da pravite
Ja sam vrlo tolerantan.
Zato bih rekao, nije u ravni. Jer ovo će se
Fantastično. Fantastično. Meni je sve ok, samo mi je. Samo mi je napako.
Ovdje je bila neka fora na ćirilicu, gdje se čita imena naših. Ona menja na ćirilici, ona prvo neću da je ovdje promeni nešto pogrešno, a sad sam hvala da prezime. Ona mislim da je nešto bilo jedno gore, sad je jedna zlatoka pored.
Dobro, da baca na 2 lejera i onda su...
Da pola. I onda je tako bilo 2 ona pojkara i on meni. I ona koji ne sam cijelo, znači kad bi ona menja možda. I ona bude rekao, te moj pomoć da sam okej, pa da će staviti, presno da se treba. Znači, ne treba mi više ja stvarno svaki sutra ne treba mi više, ja sačekam, ali od 60 miliona pada s možeš si kako ide meni, kako vas treba. Ovo sad je
Sam skoro cedilku mislim da sam rekao da sam tamo sve.
I guess we can get started. Hello, everyone. I'm Aleksandra Trajković, and it is a pleasure to host this session. The topic is ecological applications, and we can proceed with our first presentation. I invite Finja Schauman to give us his talk.
Should I use this? Sorry. No, it's no problem. Is this working?
Should work. You have to touch it down.
That's right. Okay, hi everyone. I'm Finja. I'm working at the Helmholtz Center for Environmental Research in Halle, Germany. And today I want to talk about my work on multiple sensors in bumblebee colonies to disentangle pressures on bee health. And I mean, as everyone of us knows, pollinators are threatened, and this is due to many drivers leading to the decline. And I mean, some examples are climate change, land use in agriculture, pesticides, diseases. So various drivers, they all act at various spatial and temporal scales, and they're also interacting with each other. And we need a better understanding to establish effective conservation measurements. In my project, we are focusing on climate change, land use change, and pesticide use. Climate change, for example, leads to environmental changes, for example, in rainfall distribution, wind patterns, and heat, but it also causes extreme weather events such as heat waves, and this leads to plant-pollinator mismatches or thermal stress on pollinators. Land use change, on the other hand, through agricultural intensification and urbanization, leads to habitat loss and fragmentation of the landscape, therefore to reduction of nesting and food resources. And pesticide, on the other hand, leads to residues present in pollen and nectar, but pollinators, of course, are foraging, so they're therefore exposed to lethal or sublethal concentrations. So all of these drivers have sublethal effects on pollinators that impair their survival, behavior, and health. And bumblebees, among these pollinators, are sensitive to these stressors. Bumblebees are social insects, as honeybees are, so they live in colonies. In comparison to honeybees, they have an annual cycle, so a single queen forms a colony in spring, then workers are produced, and later on reproductives, and only the new produced queens are surviving the winter. So it has been found that in terms of climate change, so in my project we are focusing on heat stress, and it has been found that with increased temperature, less offspring was produced, and in terms of food resources available in the landscape, it was found that the less flower density, the less queens were produced over the season, and also in terms of pesticide use, less queens were produced in treated colonies. But this is just one response of such colonies. There are several responses on different levels, individual and colony level. One quite obvious one in terms of heat stress, when the temperature inside the nest is too warm, they start to ventilate to cool down the colony. Other responses are, for example, different foraging activity patterns, reduced flight durations, or also inside the colony, the internal organization gets chaotic, let's say, or is altered. Also, the hatching success is reduced, or the investment into reproductives with workers is altered as well. So there are several responses, and in my project, we want to capture some of these. So therefore, I developed such a multi-sensor monitoring system to monitor bumblebees. So it's quite nice. We can order bumblebee colonies from commercial breeders. This is from BioBest, so we don't have to breed them ourselves, which of course you could do. I placed these colonies in those houses, and here you can already see there is a camera capturing images from this entrance board, and there's also a camera capturing images from above, so inside the colony, and there's also a microphone capturing the acoustic activity. And I will now go through all these different sensors to show you what it's looking like. Okay. Yeah, so the camera at the entrance, quite obvious, is placed 10 centimeters above this entrance board. And with that, I want to derive information about their foraging activity, about their flight duration. So I also individually marked some bumblebees. You can see with the white dot there on one individual. The fanning activity, which is what they're actually doing there at the moment, what you can see, and many more. And I took 4 images per second during the whole day. Then this is how it's looking like from the inside of the hive. So there I take 1 image per minute to capture their internal organization and see what kind of cells they are building, the hatching success, and so on. This is the overall aim with this data. And it's quite nice. This is like a recording of 1 day. To just look at what they are doing is also very interesting. Then I have the microphone placed inside the colony, capture the overall sound activity over the whole day. So really 24/7 over the whole season. I captured 5-minute recordings every 50 minutes using an audio muff, so also capturing ultrasonic sound. And then I also have an automatic scale that is combined with some temperature sensors inside and outside the colony to record the ambient temperature and also the nest temperature to get information about their thermoregulation success inside the colony. And the automatic scale also measures the weight every 5 minutes. So I have a lot of sensors, and now the combination between the aim, we want to detect stress in these colonies. So I did some treatment experiments of these 3 stresses: heat, food availability, and pesticide use in 2 rounds over 6 weeks. And I did first the heat and food availability treatments. Food availability here means perfect condition because they could freely fly. The treatment colonies were just fed ad libitum, so they always had food available, in contrast to the others that had really to go out and forage. And same for the pesticides. I mixed commonly used pesticides in their food in different concentrations. And after this season, I collected really a big data set, and now I'm in the state of analyzing this. And what I've done so far is I developed a semi-automated label distillation pipeline, because I mean, annotation work is very time-consuming, and automation is very helpful. So I used foundation models for this, so OLV2 and Clip, with an input of some embedding images, and then I manually corrected these annotations and then trained a detection transformer. And this worked really well, luckily. So now I have this detection model and can do some first simple quantifications of the activity at the entrance boards. So here on the plot, you can just see the average counts per 30 minutes intervals, and you can nicely see activity starts around noon, has a peak in the afternoon, and then it drops again. But this is not enough. I want to derive also behavior. So the next step now is to further label my data and place some key points for post-estimation and hopefully also for tracking of individuals across frames. And at the moment, I defined 5 key points. So the abdomen, head, thorax, and the antennas. But if anyone has suggestions or experiences, let me know, because I haven't started labeling, but I'm about to. And if you have any suggestions, if I should include more or less, I would be open for discussion. And further, I want to also group them into more groups, such as like fanning individuals, or if this is a queen, or if it has pollen and so on, like all these different classes. And also I saw that there are many possibilities of how to do such post-estimation and tracking training. So if anyone has experiences and could recommend what I should use, please let me know. Just a short overview of the acoustic data. So I played around a bit in Orange. I don't know if you know this software tool. It's quite nice to just get an idea about your data. So I calculated some FFTs and did some temperature versus control colony classification, and saw that it works perfect almost, which I mean, you have to investigate. And I looked closer and saw that I can also quite accurately predict from which colony the recording came from, which I mean could be that they are all individual and have their own dialects or something, but I zoomed in also a bit more and saw that there are some vertical lines here and several frequency bands, which I think is coming from fingerprinting of the hardware, because I see that this is different from different colonies, and therefore, I mean, the model can quite accurately predict where it's coming from. So there's quite some detective work going on, and I mean, if anyone has suggestions how to clean this data, I would also be happy to hear. And just as an outlook, I also placed 20 colonies in 4 sites of different agricultural intensities around Halle, so where we are working. And I monitored them with the same system over the whole season. And the idea would be to later apply the developed models from the treatment experiments to this data to see if we can actually detect present pressures in these landscapes. Yeah, and with that, I want to thank you for listening. And
thank you, Finja, for the perfect timing. I didn't have to squeak, but I kind of hoped I was going to get a chance, hopefully later. So I open the discussion now.
Thank you for a very interesting talk. I've actually got 2 questions. I'll try and ask them together, and if you have time to answer both, great.